Yeah, ma'am. Thank you. First of all, let me thank Vikrant sir for accepting our invitation to spend his valuable time with us and give us some insights in image processing.
My pleasure. My
Mr.
pleasure,
Vikrant
Pawan.
Bhateja is an associate professor from Department of Electronics and Communication Engineering and is currently serving as Dean for Research PPC Lucknow. He is also the Dean for Quality and Academics and Quality Control. He has 11 years of teaching experience and more than 200 plus journals to his credit. And he is editor in chief for Scopus Index Journal. He has done lots of conferences across India and Japan, Mauritius. He has done lots of conferences. Thank you so much, sir, for spending your valuable time with us. I carry forward the session to you, sir.
Okay. I just share my slides. Huh?
Yes, sir. Yes, sir. I'm able to see your slides.
Okay. Okay, if the slide is visible, I'm fairly audible, Pawan?
Yes, sir. Yes.
Okay, so I just started.
Yes, sir.
Wait. Okay, the full view is visible of this slide?
Yes, sir.
Okay. So, a very good afternoon to everyone. I welcome you all to this session. That is FDP on high quality medical diagnosis using data sciences for societal benefits. As I see the planner, in the first 2 days, the audience would have learned about the data analytics in the medical imaging, medical diagnosis, and in the field of healthcare. In the last session, Dr. Nilanjan would have covered handsomely about computer-aided diagnosis. Now, my today's lecture is on computer-aided analysis of medical images, where I'll be taking up a dedicated research case study on breast cancer detection. So, this is one dedicated case study for breast cancer detection. So, the entire perspective of sharing this entire lecture today, there are some backgrounds behind it. That first of all, many a times it happens that one kind of medical image strategies or diagnosis cannot be generalized to the other ones. So based on that, say the specificities of one particular type of medical modalities may not be applicable to the other types. So that is why the algorithms, image processings, and the kind of analytics that we are applying may vary from case to case. So this is one particular case study on breast cancer detection. But I'm sure with this kind of a case study, one can also be able to, generalize and correlate that how similar research paradigm could be made applicable to other types of computer-aided aided analysis. That is when you're talking about, say, other types of detections, like maybe lungs cancer detection, or maybe carcinoma detection using ultrasound images, or for the brain tumor detection, etc. So before we begin, little about me. My major research expertise is on computer-aided analysis of mammographic images, MRI and ultrasound images, where early diagnosis of life-threatening diseases is the major agenda. My other research aspects were where I have worked are image filtering and denoising, signal fidelity or image quality assessment, image fusion, and also biomedical signal conditioning that is applicable to one-dimensional signals like EEG, ECG, and EMG. Now, for the today's lecture, the major content resource that I have referred is basically my recently published book. This is Non-Linear Filters for Mammogram Enhancement, a robust computer-aided analysis framework for early detection of breast cancer. This is my recently published book, which has just come online and in the market in January 2020. And this is a complete research which covers discussion on non-linear filters for mammogram enhancement and how they could be used for computer-aided analysis of breast cancer. So I'm sure this book could be very helpful for those who are, budding researchers, or maybe, they are in the initial stages of their PhD and they want to, say, start their research in this particular domain. So about the today's session. Today's session, I'll be covering 3 major modules. The work has been divided. In the first module, I'll be talking about introduction to computer-aided diagnosis of breast cancer. Like computer-aided diagnosis, you all already have a good discussion yesterday, but I'll be just molding it that how this CAD system we are going to apply for breast cancer. Then what are the specificities of mammographic image analysis, that how this particular image analysis is different from other medical images. Then computer-aided analysis of mammograms. Coming to the second module we'll be discussing medical image enhancement and their associated challenges. Then histogram equalization and how it could be applied for research problem formulation. This is a very simple case I have taken, that how advancement could be imbibed in this particular approach and how it could be used for research problem formulation when it is being applied to the mammographic images. Then results will be discussed over there. And then in the last module, I'll be taking, if time permits, I'll be taking 2 more case studies related to the same. So coming to the first module, that is introduction to CAD of breast cancer. So just before we start discussing the actualities, the CAD, the image processing concepts which were involved, just let's take a quick facts that what are the no's and hows of this particular cancer. So as other cancers, this breast cancer is also one of the most dangerous types of cancer. And almost this is the second dangerous in comparison to the first one, that is your lungs cancer. Now, whenever we are doing any types of cancer related studies for computer-aided diagnosis, the prime agenda is early detection, because for all types of cancers, when they are being detected or when they are being diagnosed, the stages that they come up or they emerge, they are at so advanced stages that generally treatment becomes a rare possibility at such types of stages. So generally, cures or treatments could be only possible when early detection could be done. So you can just see this image over here. This is the image of some of the cancer cells. Now, so they are just the initiating cells, which may not be very dangerous. You can have an idea that it has a very, very circular or circumscribed kind of a periphery. Now, if you see the another image, you see, now the periphery of this particular cell is getting disturbed, and you can see the lines which have started radiating from the outer parts of the periphery. So in the first image, the tumorous or the cancer cells, they are, in the very, very initiating stages. They may be present over there, but they may not be very difficult, or they may not be very dangerous. But once you see that there are some emerging speculations or radiating lines from the boundary of this particular cell, you see that this cell has now becoming active. Now, this is projected to be dangerous, as now what has happened is that certainly this particular cell has started affecting other cells. So if you see the another particular image, now you see that this cell has now started affecting the nearby cell. So what you technically define in medical terms as that metastasis has started taking place, that one cell has started affecting another cell. And finally, at the later stages, these cells become dangerous and they start affecting the other parts of the body. They get spread to other parts of the body. So that is why we say that early detection is the major aim over here, what we are trying to achieve out of computer-aided diagnosis. So if you see the very first image, this is a kind of a tumor which is passive in nature. So you say that this is a benign tumor. But once you see that the speculations or radiating lines start coming out of particular region, then you say that the tumor has become malignant. So generally, this particular cancer is affecting the woman in the age group of 35 to 55. But the major challenge is that in the stages when the women are in 30 to 35 years of stage, these cancers they may be, say, in the initiating stage, wherever they are, but they are rarely visible. It's a totally asymptomatic stage where, no symptoms, no lumps, or no discomfort is available. It's present in any part of the body. So they are present inside, but by the time they are under full growth as a part of a malignant cancer, it's too late and they generally emerge after, say, 40 years of age. So if you want to screen this particular cancer or the breast region, the mammography is the very basic or fundamental technique which is being used for screening of such types of breast pathological disorders. But the challenge is that for analysis of these mammograms, that is the outcome of these mammography technique, the X-ray of the breast region, it is being done by the experienced radiologist. So now the entire background over here is this, that if you need effective treatment of this particular cancer, it is necessary that whatever treatment, it should be initiated at the asymptomatic stages, because once the symptoms emerge and they start getting visible, probably that stage is so late that there are very rare chances that it could be medically treated. So detection at early stages is the only paradigm over here which could be used to reduce the breast cancer mortality. Now, if we take the overview of computer-aided diagnosis, then this is the very fundamental diagram which is known to one and all regarding computer-aided diagnosis. Say you have a repository of medical images over here, then pre-processing is performed, feature extraction is performed, followed by feature selection, and then there is a classifier or a machine learning stage where you classify the particular anomaly, that say the particular region is cancerous or non-cancerous, right? Or it is not abnormal or it is abnormal. Now, this is the general CAD diagram which is known. Now we want to see the version, certainly, which is applicable to or specific to the breast cancer. So now we are going to extend this particular block diagram as if now you are going to apply it for the breast cancer detection. So for this purpose, suppose this is our first block, that is medical image acquisition. So certainly for medical image acquisition part, you are taking the images from an X-ray machine, that is the breast X-ray or the mammography system. So certainly nobody is going in for a live images or the real-time image collection from a particular device. So for this purpose, we replace it with a kind of a database where this such kind of images are already available in the repository, and they could be used for further research analysis and experiments. Now, the next particular module, next particular block after that is pre-filtering. You may require any type of, say, noise removal or any kind of filtering at the initial stage. And this particular module is known as the pre-processing module. Now, coming to the next step, that once you have the medical image, that is the mammographic image, it is available to you. You are doing pre-filtering. Now, once the pre-filtering is done, what is important, that some form of enhancement should be applied to those images. That means the contrast of those images should be enhanced. So here on one aspect is to be focused, that which is different from other kind of image processing issues. Like see, if you are doing enhancement over here, now this enhancement methodology or algorithm that is to be made applicable over here, certainly this may not be the same usual algorithm which we are applying, and we are getting very good results in case of conventional image processing, because certainly when we are going to apply such algorithms directly on medical images, we may not get that good results as we are getting it on the conventional images. So definitely, if you are talking about the enhancement algorithms pertaining to medical images, you have separate or little different set of algorithms which are being used over there, or the existing algorithms are being used, but they are used with, say, minor variations or with certain types of, say, adjustments. Now, once enhancement is done, it is necessary that segmentation of the particular region of interest can be carried out. And once the segmentation is done, an individual can go for localization of the suspicious region. So suppose we have a tumor which is say present in a particular image. So after enhancement and segmentation, certainly that tumorous region is localized for further analysis. So this particular module over here is known as the detection module. Now, after pre-processing and coming to the detection module, now it is at this particular stage, a radiologist or a medical expert can intervene over here, and that person can look into these images, do investigations, and can give results over there. But this stage is not that independent that it can predict results right now on its own. So over here, say if we are analyzing a system up to the detection module, so at the detection module, the diagnosis can be only made manually by the intervention of the radiologist or the medical expert or a doctor. Now, if we want that this diagnosis should be done in an automated manner, that say a decision support mechanism may be imbibed over there, and that system can directly classify the images into tumorous and non-tumorous. So we go for a next module. So over here, once the localization of the suspicious region is done, from here we are extracting certain features. Now, once the features are extracted, there may be extra features, there may be redundant features over here, which may increase the computational load during the processing. So over them, feature selection can be carried out, feature fusion can be carried out, optimization of features can be carried out, and at the final stage, a classifier or a machine learning tool may be incorporated, like say ANN, SVM, or say a deep learner can be applied over there, so that you get a complete diagnosis that whether a given particular region of interest is, say, cancerous or non-cancerous, or it is normal or it is abnormal. So this particular module is known as the diagnosis module. Now, I'm sure this overview, this detailing has already been widely discussed in the yesterday's lecture, but recapitulating it today is for a purpose. Now, the point is that when we are selecting research problems in this particular domain or area, see, there is a kind of saturation, because what happens that people do innumerous voluminous literature survey at this particular point, but you come to know or you come to analyze that almost everything or whatever thing you want to change or apply, you seem to see that same thing has already been done or it has been replicated. So here is, I would like to give you a slightly different angle of visualizing this whole thing, specifically from the point of view of, say, where we are starting research in this particular domain. Now, what happens that generally researchers are very much saturated if you go for, say, selection of the research problem on entire CAD module, right? Because you see that here it is a pre-processing module, here you have a detection module, or here you have a diagnosis module. So you have 3 separate modules over here, but generally most and majority of papers are there where the complete CAD, these all 3 modules are imbibed and researched under a complete paper. So very less fairs are available where one can originate the research. So here my view is little different, that sometimes what happens, suppose this is our classifier stage. Now when we are doing or replicating research on the classification stage, you go in for changing different types of algorithms. Say suppose somebody has done research using ANN, so now I may do the same by changing with SVM, or I may apply some optimizer over there, like say we apply a PSO approach over there, we apply a BFO approach over there, we apply a ant colony approach over there. Any such thing could be applied to optimize the results, right? Suppose somebody want to feature the focus on the feature extraction, people try to change the research by, say somebody has done the entire research analysis by using X set of features. Okay, I should try to do the same thing with the Y set of features, and if I get a little better or little different research results, then certainly say this may become a very good problem for my MTech dissertation or my PhD dissertation. Now there are a few things which should be analyzed, and they are much worth and could be much sphere for problem formulation, because certainly voluminous work is yet to be done in those domains. Like suppose you take an example that we are changing the classifiers, but certainly the accuracy is saturated about a certain point. Now maybe because you are turning around the same sets of images. Now if at all, just think that this enhancement module may not work properly, or that particular region of interest is not visualized in a better way, it is not segmented in a better way, there will be definitely non-satisfactory performance of the feature extraction. Because if this particular module response is not good, then definitely feature extraction, your algorithm would be very good, but certainly if the right region is not localized over there, or it is not segmented properly, then definitely you are not going to get the actual features over there. So once the actual features are not given as input to the classifier, certainly the results may not, or they may saturate beyond a particular point. So definitely one probable area of a very dedicated research could be that you should focus on the detection module, or the problem can be selected by developing new kind of enhancement filter or enhancement module, new kind of say segmentation module, so that the entire detection process can be made to give more promising results. So once the detection module is going to give you the promising results, certainly your feature extraction or classification is going to be catalyzed, and certainly there could be a jump in the accuracy. So this is one particular sphere where some way an individual may, initiate the research. Another part that people try to explore the available set of feature extraction techniques or the feature extraction tools which are available. But the point is, you would have already learned that feature extraction tools are nothing else, but they are just small parameters, or rather you say they are the statistical parameters which you are extracting from your region of interest. That is when this particular region is localized, your anomaly region is localized after the segmentation. So now there may be possible that you could have explored only certain set of statistical features for extraction. But if you think the same from the mathematical or statistical point of view, there are a lot of statistical parameters which are not even explored or yet correlated with the image processing. So certainly, if such new statistical parameters could have been experimented, tested for the purpose of, say, feature extraction of any kind of images, certainly the results may be different. So if somebody has a good statistical or mathematical background, certainly doing cohesive and more focused research in the field of, say, feature extraction, feature selection and optimization could be an entire problem in this sphere. So these are, different avenues where, an individual can, frame a complete problem in the sphere of computer-aided diagnosis. So over here we say that the entire CAD module is being classified into two types. One is known as CAD-E. So when we say CAD-E, it is basically the detection module. That is this second module that you are seeing over there. And when we see CAD-X, so that is the diagnosis module that we are seeing in the end. So the only difference is that if at the response of the detection module, that is CAD-X, we are just applying, say, a kind of decision support mechanism with the help of feature extraction and classification, it becomes a CAD-X, that is your computer-aided diagnosis approach. So this is over here. I have, just recapitulated the entire CAD thing so that, it could be very well correlated with the research problem selection. Now, let's see this entire block diagram of CAD which we have seen. Now we see that practically how does it works for the breast cancer detection. So it is just a visual to see. Now suppose we have a input mammogram. See, it looks like this, a breast X-ray image. Now this particular mammogram is, say, directly taken from some particular research database, which is, contributed or prepared by some hospital. Now, suppose we have done some pre-filtering over here, or say we have done some pre-processing over here in the form of, say, RGB to grayscale conversion. So this is how the image appears. Now suppose enhancement is applied over there, segmentation is applied over there, and now the region of interest, you can see that this is now the region of interest which has been localized and separated. So certainly in this image you see that this is a particular tumorous region or a particular doubtful region which could be, doubted as a tumor, and it, requires further investigation and analysis by a medical expert. So this is how, the CAD approach has worked, and the region of interest has been, say, localized or segregated over here. So the ROI is detected. So you see that this is our phase one, that is CAD-E or computer aided detection. Now what we have seen previously, that the response of CAD-E or computer aided detection is now given as input to a decision support system, where you get the response that is known as CAD-X. So now what we are supposed to do, we have to extract features from this particular region of interest. So what we do, see, now we have this detected region of interest, and now we are going to extract features. So let's see that we are extracting some textural features over here. So as I told you that they are, all your statistical kind of a features. So one is, you see that mean is there, standard deviation feature is there, contrast, correlation, energy, homogeneity, or it could be any other type of features. Now these features are extracted from the ROI. So what we see that now your ROI is an image. Now we are converting that image to a vector form or a number form, so that now it can be directly made input to a classifier or a machine learner. Now, out of these features, I mean, there could be certain features there which may be redundant, and we may want to, go without those features. So that is what we are doing in the feature selection stage. See that we have not taken all the features, but say on some basis of some decision making, we have selected certain features, or say we have optimized certain features. So this is what happens under feature selection. And once feature selection is done, say now you have a pool of 4 types of features over here. One is your mean, contrast, correlation, and homogeneity. So now this particular set of features, a feature vector has been formed, which is consisting of these 4 features, and they can be given to a classifier, from where you get your output in the form of, say, that whether the given tumorous region is a malignant tumor, or it is a benign tumor, or it is a normal region of interest, or it is a abnormal region of interest. So this is what happens in a computer aided diagnosis module or a CAD-X module. So that was about the introduction to the CAD. Coming to the next module, that is your mammographic image analysis. So let's take an insight into it, learning some no's and how's about what is different about mammographic images, that is the mammography images, and how these images may be different from your conventional images, how these images can be different from your, say, medical images, or say other category of X-ray images. So now, just to know, mammography is being a very effective and popular modality for initial screening of your breast cancer. So this is a kind of a X-ray system which is used for acquiring these mammograms or the breast X-ray images. Now, let's learn about certain things, certain facts over here, which you will correlate that how this image is different from other images. Now, the first and foremost point is that this particular screening employs very low dose of X-ray. Now, once the dosage of the X-ray is very low over here, the outcome is that the visualization of the details becomes very, very poor in the final image. But reason for keeping the low dosage is that this particular area, the breast region, is a very, very sensitive area. So certainly the kind of radiation which it is being exposed to, it is not as same as maybe when you are doing a chest X-ray being done, maybe you are doing the bone X-ray of, say, a particular arm or wrist or a particular leg region. Certainly those images are more clearer because you have heavy doses of X-rays which could be used over here. But comparatively, mammographic images are hard to discern because you have poor contrast, and the reason is that you employ a low dosage of X-rays over here. So just an analysis that these are the different dosages of, say, radiation dosages of X-ray which are used for different parts of body, say for the lower region, upper region, spine, and then mammography. So if you see this 0.4 millisievert, so this is the lowest possible, radiation dose which is being used to, say, screen this kind of cancerous region. Now again, there is a reason behind it, that what happens that if somebody is going in for say particular type of a screening, then initially no one knows that whether there will be some investigative finding or not. Now suppose an individual uses or by a heavy dose of X-ray is used over there, then the problem is then that X-ray dose, it may trigger the cells, and maybe because of just getting that X-ray done, the non-active cells in the region can be eventually converted to active ones. Or you can say that the initially benign tumor, which might be present over there from years without any problem to the individual or the body, but now maybe after taking the X-ray, those ionizing radiations would have penetrated that particular tumor's region, it would have activated it, and suddenly malignancy would have started percolating in that region. So that is why this is another challenge over there, that you cannot go in for frequent testing. Say suppose you have a doubt, you have gone one breast X-ray done, you want to take a second opinion, you get another X-ray done. So every time you're getting this kind of a screening or repetitive screening in say short span of times, eventually you are more being exposed to the danger of this kind of a cancer. So reason for choosing is this, that it's a low dose X-ray and it is an inexpensive breast examination. So as I told you that the doses are low. So once the doses are low, you have poor illumination over here, you have high noise levels over here, and because of this there are challenges in the X-ray detector system, and they are affecting certain attributes of the images which you are finally acquiring in the form of breast X-ray. So the major parameter which gets affected because of these noise levels, or you say that because of this low doses, is the image contrast, which becomes eventually affected in these images. Then signal to noise ratios get affected over there, because you may get a final mammographic image having a very low signal to noise ratio because of the noise levels. Then the spatial resolution is also challenged over there, because once the spatial resolution is challenged, it is very difficult to visualize the sharpness of the images. Maybe the image is acquired, but you cannot clearly see the edges over there. Say you cannot see the edges over there. Now you can correlate that if spatial resolution of the image is affected, you cannot see the edges, and regarding the cancer diagnosis, 80% of the diagnosis is being made just by visualizing the edges or periphery of the tumor region. So image sharpness is also affected when low dose mammographic X-rays are used. So because of these reasons, mammograms are among the most difficult of the radiological images to be interpreted by the radiologist, and certainly that is why specifically for analyzing mammograms you require a really skilled pool of the doctors or your medical experts. Now coming to the next that is the types of mammograms. Just you can have a view of these images. Say if you move from left to right, you see that the first thing over here, you have a fatty mammogram over there. Once the mammogram is fatty, you see there is no tissue accumulation over there. It is a very clear kind of a mammogram. So if there is proximity or tumor in any of this region, it could be very easily detected. Then you have mammogram with scattered fibroglandular tissues, where you see that the tissues are existing in certain patches over here. You can see a patch over here, like these are the scattered fibroglandular tissues. Another is, say this is a heterogeneous dense breast, where you can see the different tissues are very densely packed over here, and then you see the extremely dense. Now, if you reverse this flow the way we have seen, you can see that this extremely dense type of a mammogram is attained at a very young age, say up to 30 years of age, And if you see, you talk about this last image or the first image, that is the fatty mammogram. Over here, this kind of a mammogram is generally available, say, after 45 years of age. So now you can correlate that younger the woman is there is a very high density of tissues in these images. So certainly a tumor may be available, but it is in the background of this particular tissues. Now tissues are also white, tumor is also white. So it is very hard to discern this particular tissue at a very early age. But certainly what happens at the later stages, you see, once the lady is moving in different phases of ages, you see that now the density of the tissue has started getting thin. And once this tissue density is started getting thin, you see that there is more proximity to view the tumor over here. And ultimately, the last, at the higher age, say 45 years or 50 years, the breast region becomes quite fatty. You don't have these fibro white tissues over there. And now it becomes very, very easy to locate that particular tumor by the screening technique. So that is why we say that for the younger woman, it is very difficult to detect this particular type of the cancer. So just an example to show you certain types of these images and how they are being classified, say, on the basis of their severity. So you see that the top images, these are the normal mammograms, like where there is no anomaly which is being present over there, right? If you see these particular sets of images, now what is happening, say this one tumor region, another tumor region is being marked. And if you see both these marked regions, they have a nearly balanced periphery over there. So as I told you, once the periphery of the tumor is very balanced, very regular, very circumscribed, so in that case, it is, say, not a very dangerous kind of a tumor. Currently, it is in the inactive stage. That is, metastasis has not started to, it has not started affecting the nearby cells. And you term this as a benign mass. Now, coming to the malignancy, as I told you, that once the edges of this particular tumor region, they start getting, say, stretched out, they start getting distorted, say like the radiating lines from a sun or something like an object. So this particular kind of stage is indicative of malignancy, and you say that it is a malignant mass. So this is how one can eventually, or you can say how radiologist differs between a malignant tumor region and a non-malignant tumor region. So certainly, say, if you understand the same thing from the image processing point of view, so it is very important that the contrast of this tumorous region should be very, very balanced, that you can visualize these tumors. The sharpness of these regions, tumorous region, should be very, very good, because once the sharpness of this region is good, then only it is, practically possible that you should be able to, visualize those edges. And once the edges are clearly visible, then, a radiologist can manually see those edges, and it can predict, he or she can predict the nature of the malignancy. Certainly, if you are going for an automated CAD diagnosis, see, we are going for a machine learning approach, then it is from these edges, from these structures, shape or edge or texture features can be extracted. They could be later, fused, or they could be, optimized or further selected to reduce the redundancy part, and then they could be given to the classifier. So this is how the severity analysis can be, ascertained from these images. Now the question, why mammogram analysis is complex? So many of the points we have already discussed in the background. So just let's summarize them. So this particular image of the breast tissues is complex. Certainly because these dense tissues may easily obscure cancers. We have seen that since these tissues are predominantly white, so if our tumor region is again white and it is present in these images, a kind of say camouflaging is there. So it is very, very difficult to detect the anomaly. So you can have a view of these two kind of images. These are the mammographic images with very, very dense breast region. You can see the high density of tissues over there. So certainly there is a very rarest of possibility to detect or locate tumors in these types of images. Now, as we have seen, that even if you have fatty tissues or glandular tissues like the other types of images which were there, they are definitely better than the dense images. But again, there are challenges of, say, overlapping of anatomical structures. Say, what happens that ultimately a 3D region, I mean, the complete breast region is a three-dimensional thing, and you are trying to, image it in a planar view. So certainly there is lots of proximity that there could be overlap of many anatomical structures in the mammogram. And certainly because of that overlapping, it becomes very difficult to analyze these images. So you can see a visual example over here that, say, if a tumor region is present in this fatty breast, you see a good contrast is available and cancer can be easily seen. But again, if the same tumor is projected to be present in the background of these, say, glandular tissues, that is white on white, so it will be really difficult to discuss detect this kind of a cancer. Again, the mammogram analysis is dependent upon the subtlety of the breast abnormalities. So another point is that this analysis is difficult because, say, right now we are just naming it by the name, say, tumor or non-tumor. But if you go to the biological terms or if you go to the radiological terms, you are classifying these tumors technically as the name of lesions, and you have variety of different types of lesions, different shapes of these lesions which are being present, and each particular lesion is, say, communicative of a different type of investigation or a different types of diagnosis. So because of these variety of abnormalities which are being present, say, for discussion or, say, analysis, it becomes difficult to analyze these images. So you can see the image of a mammographic mass, right? So these are the dense region of different sizes which are available over there. But generally, if the tissues are glandular or dense, it is very difficult because they are camouflaged in the background of your white tissues. So here you can see the different category of masses, mammographic masses, which are, say, available. So if you see the first category is the circumscribed one. You can see the balanced periphery of or edges of these images. But again, as you see that there are radiating lines coming out of these regions. So these are your speculated masses, and say they are more prone to malignancy. Circumscribed masses are generally, say, they are non-cancerous, or mostly, with few exceptions, they are benign in nature, right? Coming to a third category over here. So this third category of lesions is generally known as the ill-defined lesions. So if you see this kind of structures over there, you see that this could be rarely classified into a circumscribed one or a speculated one, right? But because of, the distortion in the structure of that area, this particular area, say, if you cannot say that this is a malignant thing, you also cannot say that this is, a non-cancerous thing. So what is important, that this particular area needs further investigation. So now this particular example will certainly give you a very good food of thought, because what happens that almost maximum researches that are being done on CAD approaches, people try to just focus on two types of abnormalities. That means ultimately your results are either being classified as cancerous or non-cancerous, right? But there is also a third category where, the image cannot be truly a cancerous one, or it may not be truly called a benign one, but it is a third category rather, which needs further investigation or analysis. But as if you are designing a conventional way, you are just designing, a binary output kind of a machine learning tool. So over there, you are just having two types of responses, that is say cancerous and non-cancerous. So certainly because of that, what happens that these ill-defined structures, they sometimes get misclassified as benign. So once they get misclassified as benign, what happens that they may be left, untreated, and they may later grow up as cancer. Sometimes, they get misclassified as malignant. So if they are being misclassified as malignant, maybe they may not be so difficult, but a doctor may recommend that the individual should go for a further advanced stage of screening, that is say known as a biopsy stage, right? And say at the cytology or the biopsy kind of a stage, it's a very, very, painful process where say a needle is inserted for the tumorous region and that particular say flesh area is being taken out for further analysis under the microscope. So certainly a patient is just on false grounds being exposed to this kind of a say invasive discomfort. So what happens, the two types are very clear, circumscribed and speculated, but the ill-defined category is something, which needs further analysis. So these are the different types of, growth patterns from which, a benign cancer gets converted to the malignant. So if if somebody works in this particular domain for further research on CAD, there are different types of mammographic databases which are available for research. And if you see different types of databases, there they have named those particular images with these particular types of say masses or tumors, that the region is totally round, or say it is oval, or once the say border starts getting distorted, say it becomes lobulated, later it converts to nodular, and once it is nodular, it converts to a star-like structure, that is stellate, and then say after that this speculation start growing over here and metastasis start. So, so if we are trying to do a shape analysis or classification of say mammograms on the basis of shapes, right? So probably there are these 5 types of shape cases could be there for such kind of problems, where you can see that the first 2 cases are generally classified as the benign one. These 2 cases are probably malignant, but again there is a third category, right, which needs further investigation. Now, this was what we have learned about, say, different types of tumorous region or the mammographic masses. Now, this is not yet over, because you have another type of, say, mammographic anomalies which are present, and they are, more smaller in visualization. They are nothing else, but if you can say very finely see, there are some small dots which are present in this area. So these small dots, white dots, are basically the calcium dots which are accumulating in that region. These calcium dots are known as microcalcifications. So once these small dots are at distance apart, they are scattered. Here you see that a kind of clustering of these dots has taken place. So this becomes a grown cluster of microcalcifications, and say at later stages, this cluster gets converted to a mass. So certainly you are detecting using CAD to detect masses, it is a good thing. But again, once a mass is formed, that means the tumor has already initiated over there. So if we go for a much enhanced early diagnosis kind of a thing, then it is more desirable to identify these calcifications rather than identification of these masses, because once the calcifications are detected at the early stage, that means the infection or the cancerous cells have started accumulated in that particular region, right? So these microcalcifications are tiny calcium deposits, and they could be obscured within the normal breast tissue. So again, you see that if you have white tissues over there, so detecting a mass at the background is a problem. So I mean, detecting these white dots again in a white background is again becomes a very, very difficult kind of a thing. So now with this, you can, try to correlate and ascertain that if some image processing kind of a thing is being applied over here, or the kind of image processing algorithm which is applied over there for visualization of these type of anomalies, certainly it cannot be just a known simple or conventional image processing algorithm that could be directly applied over there. You definitely need some specialized touch, in the algorithms which customize that particular algorithm so that it can highlight or visualize these type of abnormalities. So this particular image is just a demo to show you how different types of calcifications are present, and initially you see that they are scattered ones, and say at this particular stage clustering is being done over there. So again, the chances of having these microcalcification is more prominent in the younger woman than at the later stages or the higher age woman. Now the impact is that because of these problems, because of these challenges of the mammographic images, there is always a difficulty factor which is present with the radiologist in discerning these images. There are psychophysical inconsistencies, like somebody is, seeing and analyzing the same sets of images daily. So there are, psychovisually 10% by default chances of error in that. So on account of that, sometimes normal regions are being classified as tumorous regions, and there are higher incidence of unnecessary biopsies. Like had it been a very normal examination, it was fine. But biopsy, as I told you, say is a very, very painful kind of a process. So just getting it done in an unnecessary manner is a very, very much trauma to the person which is facing it. So another process to go for increased accuracy is for double readings. But again, if you are taking opinions from two radiologists, then they may contradict, and the cost factor is also increased over there. So accuracy is definitely a challenge when you are framing the problem of CAD for breast cancer, and so computer aided diagnosis is therefore a preferred solution over there. So once you see computer aided analysis over there, so computer aided analysis, we see that the same image is being analyzed on the computer screen by the medical expert, just to, so that, they can zoom in a particular area, they can visualize a particular area, they may omit a particular area and then see it. So there are software based tools over there which are embedded to, visualize these type of images on the computer screen. So if you want to empower this computer aided analysis, certainly good visualization algorithms have to be applied over there. Good enhancement algorithms have to be applied. Certainly the contrast, so that the contrast of those particular regions can be improved. Not only contrast, but say the sharpness of those particular regions could be also improved. So these are all the background requirements when somebody is trying to frame the algorithms for computer aided analysis of mammograms. So with the sole aim is that if this particular algorithm is going to yield good performance, certainly you have a better segmented region. And once that region of interest is more accurate, more precise, more visually clear, then the CAD-X system or after feature extraction or classifier, the response are more prone to be more accurate, as it has been, without a very good enhancement approach. So therefore, once you want to go for a computer aided analysis of mammograms, this could be only possible if you have a strong mammographic image enhancement algorithm. Because if this particular algorithm is not good, you're not getting a good contrast, you're not getting good sharpness. So in this particular cases, you cannot go for a early diagnosis. So now the medical image enhancement and its challenges. So we know that, just a quick review, image enhancement is, where you are trying to improve the contrast of a particular image, so that, trying to maximize the difference between the maximum and the minimum pixels over there. You are trying to do a amplitude rescaling of each pixel over here so that the contrast can be better. Another aspect is that when you talk about contrast enhancement, there is another aspect known as edge enhancement or sharpening. So that is why if we apply this concept to the medical images, so we want contrast enhancement, and we also want to have edge enhancement, because until and unless we are having edge enhancement, sharpening is not obtained. Now, let's talk about medical image enhancement. So medical image enhancement is that you are deploying the image enhancement algorithms, which are your mathematical tools via which you can control or improve the contrast of the particular image. One can enhance the noise component which is present. One can, enhance the noise component also in that image, which could be undesirable. So the problem is that if you are using conventional image enhancement algorithms, then certainly sometimes the contrast improved is not very good. Sometimes it may, increase the usual brightness of that image, that, the usual look of the image also get tampers. Secondly, sometimes in the aperture to increase the contrast of the image, what happens, the algorithms tend to falsely, increase the noise component in that image, which again creates a problem. So you want all those such types of techniques for medical image enhancement, where, contrast can be improved in a balanced way, and also there is no undesirable enhancement of the noise during that process. So that means when enhancement is being done for medical images, one has to ensure that in the venue to improve the contrast, to amplify the contrast, one should not go in for amplifying the noise component over there. So if we talk about mammographic enhancement algorithm, so now again, conventional enhancement algorithms cannot be very good for mammographic enhancement algorithm. So when you are talking specifically about mammographic enhancement algorithm, we are trying to improve the contrast between the region of interest and the background. So say the point is that suppose, say this is your mammographic image, and over here, say a particular tumor region is existing. So now if I want to enhance this particular tumor region, I may not have to take reference of the full image. I may just have to take a nearby region of this particular image, that is your region of interest. Now, certainly, what I am supposed to do is I have to apply the algorithm so that the contrast of this foreground, this is the foreground region, this could be enhanced. Now, if I somehow want, say, to emboss a white region, one way is to increase the brightness. But the other way is that if you place a black background at the back, then certainly the white region is more embossed. So it is not just, that to improve the contrast of the foreground, but what is important is that one has to also suppress the background. So sometimes, improving too much contrast of the foreground may increase the noise of your image, right? It may hamper the signal to noise ratio of the image, but rather than, applying so much stress on the increasing of the contrast of the foreground, one may go for the suppression of the background also, so that, tries to suffice and eliminate the effect of the noise enhancement. Again, sharpening of the lesion edges is again the requirement from the mammographic enhancement algorithm. So if you see that this is a regular image, over here, this particular region of interest has been extracted. Now, if you say apply a conventional algorithm to it, you compare these two images, there is no much change as far as these two images are concerned. So this is the original image, and say this is the outcome of a conventional image processing algorithm, right? Now suppose if you try to say increase the contrast beyond a point, you see that the noise is enhanced. So how do you see that noise? So you see that the brightness of the region has unnecessary enhanced, which is, you know, trying to disturb the edges over there. So this is how, what you call that, when you are enhancing the contrast, noise is enhanced. But if you see this last image, what you see, I have not certainly improved the contrast much. The contrast was white and some part of whiteness is increased, but rather than now, you know, increasing the contrast of the foreground or whiteness of the foreground, what we have done is we have suppressed the background. So you see that it is the suppression of the background that it has made the sharpness increase. You can see that the edges have become very, very clear over here, which are the predictiveness of the malignancy. So with this kind of enhancement algorithm, if such type of enhancement algorithms have been applied to medical images, they help to improve all the outcomes, you can say all the problems which are due to the malfunctionalities of the X-ray detector. It is improving the contrast and it is also trying to improve the effective spatial resolution. So when we say mammogram enhancement, we see that we are enhancing the contrast and we are also enhancing the edges over here. Now, another part is, as I have already told, that sometimes we want to, in the too much urge to enhance the particular image, what we are doing, we are increasing the brightness of the region of interest. So this excess brightness also becomes, you know, the entire image useless over here. And this particular attribute is known as what? Amplification of unwanted noise. So again, you have to improve the contrast, you have to improve the edges, but certainly both these processes should not increase the noise in those images. Now suppose noise is present in that image, then, you know, a suitable solution that may appear to anyone is to apply conventional noise suppressing algorithms. So you see over here, if say this is your original image and you see the noise appears in this image. So if you apply a conventional noise suppression algorithm over here, so what you see in this last particular, what you see in this last particular image, that the edges have become blurred in this particular region. So this is the outcome of your, say, if you are trying to filter out much on these images, just to, you know, remove or, you know, filter out the noise, you may do that, but that may be at the expense of losing the fine details for your diagnosis. So now we come for a new kind of a conclusion over here. So that if we are talking about mammogram enhancement, it is just not about enhancing the contrast. It is not just about sharpening the edges, but it is also about noise smoothing. So when we say mammogram enhancement, try and remember that we are trying to develop an algorithm which has all three aspects. That is, it should improve the contrast, it should improve the sharpening, and it should also suppress the noise during the process of enhancement of contrast and the enhancement of edges, right? So there is always, you know, a compromise between noise smoothing and contrast enhancement. There is always a compromise between noise smoothing and edge enhancement. So you can just see the same thing with the example that suppose this is the original image, this is the original region of interest, and suppose we want to do excessive contrast improvement, what we see that the overall brightness of this particular region has increased, right? So again, the contrast enhancement should be noise controlled. In the similar way, if you see that we are trying to improve the edges of that particular region too much beyond a point, then you see that the edges are also excessively enhanced and they are appearing to be blurred, right? So these are two constraints where we see that contrast enhancement should be noise control and edge enhancement should be noise controlled. So the conclusion is that one just not go for a contrast and edge enhancement, but we say rather there should be noise controlled contrast and edge enhancement. Another thing you can see over here, that suppose we have this image. Now this may appear as a normal image, but if you see the original part over here, say few dots are appearing, and say certainly a microcalcification cluster is present over here. If you apply say a normal algorithm over there, say there is no much change in the intensities, and again the image is unclear. Now if you again try to apply say enhancement to a greater degree over here, again you say over brightness has come in this images, which is again affecting them. So detecting of masses or tumors is difficult, but detection of microcalcification is much more difficult, because it is generally in the background of the dense tissues, or microcalcifications are more prominent in the younger women, where the background tissues are generally dense. Now again, an important thing which you should know, that is known as the image restoration challenge. So if you see this image restoration challenge over here, just try and correlate a very important thing, which we may not have, I mean, it should be known to everyone, which may not have, been focused so far. See what happens. Suppose this is the original image, this is the noisy image, and this is the restored image. So what happens, suppose once restoration is done, that means, I have removed the noise. So if I want to check back the quality of this restored image, what I will do? I will certainly compare this restored image to the original image, because my original image was very, very clear, and say this is my restored image. And when I compare it to my original image, I can certainly miss out that say 80% restoration has been done, or say 70% restoration has been done. But now say if we apply the same concept or the same thing to the medical images, or say the mammographic images, now you have an original image over here, which is a mammogram, and now you have a restored image, which may be, the improved version of that image. Then certainly it becomes very difficult to compare that improved image from the original image, which was actually a not improved version, or it was an inferior version of that image. So this is the prominent problem that we face while you are analyzing the quality of the medical images, because your initial images are not that good that they could be treated as reference for your experiments. So this is the reason that say this is one image and this is say the second image, and if we say that the second image is the improved image of the first image, so it is very difficult, that you compare it with this first image and then you ascertain the quality. So now there are different types of mammographic enhancement approaches which are, related to your simple image processing approaches. So say they are your histogram based approaches, unsharp masking approaches, non-linear filters, or you have multi-resolution processing where you have wavelets and contourlets. So now each of them is a kind of a separate case study if it is, applicable to mammograms or any set of medical images, right? So in the next module, I have taken up, and related the entire this mammographic work of computer-aided detection by this simplest possible approach of the histogram-based approach, because, certainly this particular approach is generally fundamental clear to all the person, who have done basic courses of image processing. So now, regarding the histogram equalization approaches for enhancement of medical images, just let's take a quick review. Now, histogram equalization, as we know, that suppose we have a medical image over here. Initially, you see that this particular image is darker in certain aspect, but once the histogram equalization is applied, it has become much brighter over here. So it is just a simple way to improve a dark image. And the idea is that suppose I have a histogram of this particular image. Now you see that the histogram of this particular image is being focused, or it is narrow to a certain types of pixels only. But with histogram equalization, what we have done, we have tried to just spread the spectrum of these pixels, and you see that the resultant image has become brighter in comparison to the previous images. So this is what happens in your traditional histogram equalization when you apply it to the medical images. Now, let's see how does it happen mathematically. Now, mathematically, what happens that, say, suppose if this is the input image, we are getting this particular output image, then what is happening that there is in the background, say, how does this histogram equalization algorithm works actually? So in the background, there is a transformation function which is present. So this transformation function is basically used to transform the simple histogram by spreading the pixel values. So you see that how this is mathematically happening is that you have an initial histogram, say, which is very narrow, and you are giving these pixels to a transformation function, some certain mathematical function, and this is how your histogram gets spread out. So this is the mathematical background of your this histogram equalization. So now it is just a kind of transformation function which is trying to recover some of the apparently lost content. Now, if we see this histogram equalization function, it applies a background function, and this background function is no other function, but it is a sigmoidal function or a logistic function, which this histogram equalization applies to all the pixels to improve their brightness, or say to improve its visualization. So you see that this is the particular transformation function which is being used for histogram equalization, and this is the basic sigmoidal function which is used over here. Now just try and recall that whichever transformation function which we are using for histogram equalization, or say it is your sigmoidal function, a very important property of this particular sigmoidal function is that this function has to be single valued and it should be monotonically increasing. Now, what happens when you apply this kind of a transformation function to the medical images? Now, the problem is that when conventionally you are applying this histogram equalization over here, you can see it leads to over enhancement. That means, say, it was good if contrast would have increased for this region, but you know, this part was already bright and this image has become little extra bright. So histogram equalization has tried to, you know, eventually distribute the brightness, and which has led to the over enhancement in those images. So you are trying to do enhancement, but it has ultimately made that image little unclear. So this aspect you can see that suppose you have an image and it has been made extra brightened. So due to the saturation of these brightness level, you see that this over brightening is known as noise in these images. So this noise is nothing else, but this noise is due to the over enhancement of the pixels. So now a very well remedy to histogram equalization is, we all know that one has to use the adaptive histogram equalization. So with adaptive histogram equalization, what you are trying to do, you are just trying to, you know, make the parameters of histogram equalization little bit controlled so that, you know, the output should be in a desired manner. But again, you see that if say this is our original image, this is the histogram equalization image, this is the adaptive histogram equalization image. So what you see that when you apply this to your medical images, what is certainly happening, that histogram equalization image was bad, it was not a very pleasant one. And certainly if you are applying adaptive histogram equalization over there, then too the image has not, given a very promising visual outcome. So that means there is some aspect in adaptive histogram equalization which is also missing when you are applying it to the medical image. So somehow adaptive histogram equalization is not giving you desired results when it is applied to medical images, or say it is applied to mammograms. So this is another example for that. So now here starts the actual, problem formulation. Now suppose I have taken this as a problem, research problem, that I need to improve the performance of histogram equalization so that, this could be applied for mammogram enhancement. So let's see how we want to move about it. So we want an improvement in adaptive histogram equalization so that it can be, applied for medical image enhancement. Now, the requirement is that if you want to improve the, say, performance of the adaptive histogram equalization, or you want to improve the response for medical images, what is necessary is that certainly there is a transfer function, the sigmoidal function, which is working on the background. And that might be the cause that it is giving you non-satisfactory result. So certainly if we think that if we try to change that function with, say, some different transformation function, maybe we can go for a better performance. So, this is just food of thought that, sometimes we just get restricted that this is our known approach. This is, say, histogram equalization, but say this cannot be improved further. Certainly, you think from that angle that maybe from the front end that image appears that algorithm cannot be improved further. But say if we try to internally change the working algorithm of that image, how? By, say, changing that existing function with some another non-linear suitable function, maybe you may go in for, say, improvement of this process. So this is how one area where, the research problem can be selected in this domain. So now let's see how we apply the mathematical logic over here. So if you see that this is the plot of a non-linear transformation function for histogram equalization. If you see this particular plot, what is happening? This is a sigmoidal function, and we already know that the requirement for histogram equalization functions is that it should be a continuous function, it should be a mathematically monotonically increasing function. So now we have to think of, say, a certain kind of a function which is, non-linear as your sigmoid function. It should be continuous in nature and it should be monotonically increasing in nature. So this is what attribute we are going to improve for, say, that non-linear transformation function for histogram equalization. Now, we see that this particular function should be single valued. Once it is single valued, we see that the inverse transformation does exist. This should be monotonically increasing function, because if it is a monotonically increasing function, you can preserve the gray levels over there to a very good extent. So now let's see the plot of non-linear transformation function for histogram equalization or adaptive histogram equalization. So this is the kind of non-linear transformation function which is required for a adaptive histogram equalization. And again, we see that this is a continuous function. This is a monotonically increasing function. Now, why this particular function is being selected for non-linear transformation function for adaptive histogram equalization? Now, see, you see the reason. If you see that in this particular function, there is a kind of a threshold that is existing. Now, what this threshold does? This threshold tries to control over enhancement and under enhancement. That means if suppose that pixel lies below this particular threshold, it may go in for enhancement. But suppose if the pixel lies above this threshold, it do not requires enhancement, because then that means that pixel is already bright and it should not be further enhanced. So, you know, this kind of a thresholding is mathematically imbibed in this function so that, you know, you can control the over brightening of the tumor region, which is applicable in these images, as you have already seen. So we see that this function is also a continuous function, which is very similar to the sigmoidal function. And if you see mathematically, we can plot this function by, say, just, you know, you are just trying to combine two sigmoidal functions. You see what you have done over here, that if two sigmoidal functions are mathematically combined, we are having this, the connection part, the middle part, which is, you know, operating as a threshold region, so that your pixel enhancement below and above that particular threshold gets controlled. So, you know, this is how we have tried to change the function of existing histogram equalization to this modified function, which is, you know, nothing else but, say, this is combination of two sigmoidal functions. Now, this is just equivalent to two identical sigmoidal functions which are bounded together at points x is equal to, say, x1, or say at x is equal to 0. So this is how this mathematical function looks like. So that you see that this is a algebraic combination of two sigmoidal functions that are existing over there, but with a slight difference which is there. That is, one point, say if your x is your input pixel, so one where we are doing minus b, and one time you are doing plus b. So with this b component, you are trying to control the threshold of the overall process, and you see that there is another component or another variable which is being improved or included over here, that is known as k. So k is multiplying the pixel values. That means, you know, it is trying to amplify the contrast of this images. So over here, if you see, the B region is known to control the threshold, and K region tends to control the enhancement. So K and B controls enhancement and threshold values. And over here, if you see, there is another component which is known as A. So this A part or A component is based on, say again, the values of K and the values of B. So that means this is not a direct constant, but this is a adaptive constant because it is depending upon the chosen values of K and B. So this is how, you know, the conventional function of the histogram equalization or adaptive histogram equalization, say, can be replaced by a new kind of a non-linear transformation function. So this you can compare like this, that this was your, the black one was your original, you know, function, and say this is how you have combined two sigmoidal functions and you are getting this new function which we are going to use for as a non-linear function for adaptive histogram equalization. So now let's see that once we have developed this particular algorithm, let's see what happens when we apply this on the experimental front. So on the experimental front, if we want to start research, so I told you that one has to start with some kind of medical databases or the research repositories which are existing. So these are the very known and common benchmarking databases which are available for mammographic research. So they are your MIS databases, DDSM databases, and the B-screen databases. You can see that this is the most common link, and going to this link, you can locate these databases and many other, locally accumulated databases, which are being accumulated as a standard databases in this particular research repository, and they can be used for experiments or simulations on these kind of works. So the results which I am showing you from the case study, I'll be just focusing on the results which are of the MIAS database, that is the Mammographic Image Analysis Society database. And over here, generally the database comprises of 322 digital mammograms with different types of background tissues, different types of abnormalities, and of different severity of abnormalities, that you have images which are benign, malignant, you have images of different masses, different calcifications. So we have to, take a sample size of images where we are, doing testing on each type of images to, to show the versatility of your particular algorithm. Now, if you talk about the pre-processing part which is being done over here, so pre-processing part, only image normalization is done. Then say with the cropping part, the region of interest can be taken out over here. Now, once we have taken the databases, there is another aspect known as image quality assessment, because certainly we should have parameters which are available so that you check the quality of images in numerical means or by quantitative means, that how a particular algorithm is responding. So over here, there could be n number of image quality parameters which are available, but two standard parameters that we are taking right now is number one, contrast difference parameter, and number two, peak signal to noise ratio component. Now, if you take these two components, in one, you are taking the difference in contrast between the two images. That is, you are taking the contrast of the first image, you are taking the contrast of the second image, you are subtracting the two to get the contrast difference. So if you have a better value, that means the contrast is improved. Now, how you are calculating this contrast? So for calculating this contrast, you have this basic formula that is, this is equal to Fmax minus Fmin upon Fmax plus Fmin. That means you are taking the highest pixel value, subtracting it to the lowest pixel value, then again divided by the highest pixel value plus the lowest pixel value. So this is how you are taking the contrast of your original image, enhanced image, and you are subtracting to get the contrast difference. So higher the value, better is the value of the enhancement. Now you see, we have also taken another parameter that is known as peak signal to noise ratio. Now, why we have taken this parameter is that certainly we want to ascertain over here that if we are doing contrast enhancement, then that particular algorithm is not improving the noise, or it is not enhancing or amplifying the noise in that region. So whenever you are calculating contrast difference, it is better to have some parameter, by which you are estimating signal to noise ratio over there. So if this signal to noise ratios are, preferably high in these regions, then you can ascertain that the contrast is improved and the noise is not enhanced. But certainly if the contrast is improved and you have a low value of PSNR, then that means that this contrast enhancement is done, but it is at the expense of, say, your poor signal to noise ratio. So these two parameters we are going to use on our results so that we calculate their quantitative values, that is IQA. So once we have taken an input mammogram, we have applied pre-processing, we have applied our new enhancement algorithm over there, we have also applied our old conventional enhancement over there, and then we have calculated the value of PSNR and contrast difference. So you can see the results which are calibrated over here. See that testing has been shown on different types of, say, mammograms. That means all types of mammogram images are being tested over there, be it fatty one, be it fatty glandular one, be it dense one. So we have taken few images that are malignant, we have taken few images that are benign also. Now for these images, say we have applied the conventional adaptive histogram equalization algorithm. Say this is the value of PSNR in decibels and this is the value of the difference in the contrast. Now on these images, again we have applied proposed enhancement approach, that is nothing else but this is your improved logistic or the sigmoid function, that is the combination of two sigmoids that we have just now calculated. Now we applied this thing. So once this is applied and we have again calculated say the PSNR and contrast difference, now what you see that say suppose for this first image, you see that there is a considerable increase in the value of peak signal to noise ratio. Now, again, you see there is also a considerable increase in the value of the contrast difference. So you can verify this aspect in all the images. So this gives us a very good inference at this stage that this enhancement approach, that is this new kind of a logistic function, is trying to give us better quantitative values in comparison to the existing one. So once we have done this, let us now say apply this on the images. Now see what is happening. This is your original mammogram. This is your pre-processed ROI after pre-processing. And now we have applied this new function over here. And this is the enhanced image that you're getting. Certainly the PSNR is 66 dB. Now you see another image. This is the original image. This is the pre-processed image. And this is the enhanced image. Now there is a catch. Catch is this, that if you see both these images over here, there is a change in contrast, but the contrast change is not very, very good. But the challenge is that we are getting a very good value of peak signal to noise ratio. We are getting a very good value of contrast difference, but visually that image is not very, very promising. So certainly there appears that something is missing over here. Maybe while compiling these results, some part is missing over here. So once, I mean, this is just to give you an idea that sometimes it happens, we are doing preliminary analysis, we are working on those results. The results are quantitatively very better, but the images are not very, very pleasant. Then this gives you a very important suspect that you are supposed to reconcile everything now. There is something wrong over here. And to test about what is wrong over here, certainly just see that you had a sigmoidal function. This sigmoidal function was dependent upon two parameters. One was the threshold, which you were referring by B. One was the enhancement factor, which you were referring by K. Maybe possible that you have not taken the right combination of values for K and B. That is why the algorithm is not giving you desirable results. So what is to be done now is, that is tuning of parameters. So now if we do little tuning of these parameters, that means we run this algorithm again for say different types of range of these parameters, and you see that where the peak is coming. So you will come to know that if we see the threshold part, then certainly your peak is coming over here somewhere. So that means, say suppose if the peak value, that is the threshold, is chosen around 0.9, it may give you good results. Similarly, if you see the rate of enhancement K or the amplification factor, you see the peak is coming over here. That means say if we choose a value somewhere between 5 and 10, you may get a good result over here. So this kind of, simulative analysis for tuning of parameters has been characterized, has been applied on all these images, and you come up with this kind of, say, tuning parameters. a good analysis comes out that suppose maybe when you have a fatty mammograms over there, the rate of enhancement should be chosen in the range K8 to 10. Say the threshold should be chosen in this range. Certainly, if the images are dense, maybe you require more enhancement over there. So here you see that the values of enhancement should be in the range 10 to 15, and this should be the value of the threshold. So now with this kind of a thing, we are going to apply our transformation function on different images. So maybe when the fatty images are being processed, you are applying the transformation function with different values of K and B. With other category of images or dense images, you have the same transformation function, but different optimal values of K and B. So now what we have done, we have tried to manually tune these parameters to certain optimal range. Now you see the results over here. Now the same algorithm which was initially giving unpleasant results, now you see the change in the result once we have tried to tune in the parameters. So if you see over here, this is the original mammogram. This is our region of interest. Now on this region of interest, if you apply histogram equalization, you get this kind of image. You see over brightening over here, over enhancement over here. Now if you try to control this with conventional AHE, that is adaptive histogram equalization, again the results are not pleasant. But if you now apply the adaptive histogram equalization with the new function, the new nonlinear function, you see this is the result where you see that this foreground region, tumor region is enhanced, and now this background has got suppressed over here. So this region is becoming more embossed for the analysis. And it is very, very clear that although you are calling it a circumscribed mass, but you see that there are some distortions in the periphery or the margins of this particular tumor, which is a very strong indicative that some metastasis is already started in this tumor region. So maybe initially it has been called a circumscribed mass, but say after enhancement, you can say that this is a circumscribed mass, but it requires further investigation, and there are some indicators of malignancy over there. And now this image can be used for better feature extraction, and then it can be used for classification. This is another view. You can see that again, you have original image, this region of interest. This is the response of histogram equalization, AHE. And now if you change the function, then say this kind of the proposed function. With the proposed function, this kind of results are available. Now again, we have changed the tuning parameters. So once we have changed the tuning parameters, again we have tried to assess the quality. Again we have tried to do the image quality assessment. So again we have calculated PSNR and contrast difference. But now if you focus on these values, there is again something very, very alarming. Now what has happened? You have chosen seen PSNR. With the existing approach, it is 54, and now the proposed function is giving you 10. Contrast difference with the existing function was 0.2, but with the proposed function it is 0.3, which is just a minor change. So now again something is wrong. You see, now the images are very, very well tuned. The functions are tuned. The images are visually very clear, but we see that now quality assessment is not giving you good results. So certainly, this problem was done by my scholar, and it took us a lot of time to reconfigure and get out that what problem is coming actually over here. And to your surprise, let me tell you what was there. Certainly, we have chosen contrast difference as a parameter. Now, we chose a different parameter over here, which is known as contrast improvement index. Now, in many papers, if you read, you'll certainly get to confuse that contrast difference and contrast improvement index are very, very similar things. But there is a very fine difference between the two and a very interesting one. Let's understand. Now, if you see, the formula is that contrast improvement index is that you have C of your processed image, that is the contrast of the enhanced image, to the contrast of the original image. Now, how you are calculating this contrast over here? Now your formula is F minus B upon F plus B. So again, you see that formula was, contrast was F max minus F min upon F max plus F min. So the formula is same, but there is slight difference over here, that over there you were taking the maximum pixel values. Over here you are taking the aggregate values. This is one change over here. Another change is, with F we mean foreground, with B we means background. That means, suppose if we have a tumor region, say this is your tumor region and this is your region of interest. So now I am not calculating contrast for the full image. I am just calculating, say, C for this region, which is our foreground, and this region, which is our background. So, you know, this is a very small change of concept, and this is known as simultaneous contrast. So simultaneous contrast is simple thing that if you are trying to, you know, visualize this particular small square in this white background, it is not good in contrast. You can see, if you see over here, it may not be good in contrast, but if you see the same square in a suppressed background, it appears very good in contrast. So it is this concept of simultaneous contrast which has been replaced over here when you are calculating contrast improvement index. And now you see that if we apply this particular parameter on our images, you see the change in values. So you see that suppose we have this particular image, now histogram equalization is giving you negative results, because actually histogram equalization is not improving, it is spoiling that image. Simple AHE is not giving you a visually clear image. You see that the contrast improvement index is very small. But if you see the final value over here, you see a very much high values of contrast improvement index are obtained, and see these high values are up to 20 or 24 that you are getting over here. So now our algorithm was correct. Only problem was that we were going by the standard parameters, and those standard parameters were not very good enough for the medical images. So for mammographic images, it is to be noted that instead of contrast difference, the chosen contrast parameter should be your contrast improvement index, because it works on the concept of the simultaneous contrast. So you can see that for the different comparative algorithms, with the red line you can see that there is a very high value of contrast improvement index, which is obtained with this kind of a function. So inferences over here are this, that your contrast difference values were not very good, but once you replaced it with the contrast improvement index, those values have changed and they are telling you the right assessment, right? And this particular function has given you good gray level modification, and there is a threshold parameter over there which has helped to control the over and under enhancement over there. So this is the biggest inference when you are using this particular function. So now for continued study, this particular paper of mine, this is a robust polynomial filtering framework for mammographic enhancement. This polynomial filter framework, this paper is being published in IEEE Census Journal, and if say a more continued study on this subject is required, this particular paper can be further studied for more advancement. Now other thing, here we have just developed this function and we have seen that it has given us a very good response on the medical images. Now you see how this same function we can apply on other aspects of research also, or other research problems also, again with the medical imaging, We are just changing different image processing algos. So this is again one problem where, you know, one of my student is already working on that. You are aware of the conventional approach of unsharp masking. Now, what unsharp masking does, unsharp masking simply tries to sharpen the images by adding a high pass filtered image to its own. So if you see the block diagram, I have an input image. I take the high pass filtered image. When you get a high pass filtered images, your edges are enhanced. You are multiplying that particular image by a gain factor, and you are again adding it on the same image just to, you know, emboss the edges. So the approach is this, that I want to make the image more sharpened. But when you are using conventional unsharp masking, then there are two problems. One is the noise may enhance, and the another is the high contrast regions may occur over there. That means there should be there are over enhancements, there are over brightness in those images. Now this is the function, your basic function for your unsharp masking, and if you see that this is your input image, then this is the enhanced image for your unsharp masking. So you already see that this over brightness is the cause of your high contrast or the noise which is enhanced in these images. Now if you see the other modifications of unsharp masking which are being made, again with these other modifications of unsharp masking, you are not getting a very valued or a differentiable kind of a response is attained. So how these modifications are now done, this linear high pass filter is being changed by some rational filter. It could be changed by a cubic filter, or this could be changed by a adaptive filter, right? So when these modifications are done, again there are constraints that you have undesired overshoots over there, you have noise enhancement over there, and you have problem of tuning of parameters. So a very good domain to change the function over here is that, suppose what change we can do is that in this conventional unsharp masking algorithm, we should replace the high pass filter by this non-linear transformation function. I mean the sigmoid function, the new sigmoid function, which we have taken up in the initial part of our preliminary results. We can add it to the conventional block of your unsharp masking. We can replace it by the new or your improved sigmoidal function, and there is a conventional change in the results which are seen. So if you see that this is a particular image where you see that the calcifications are present. If you see the response of adaptive histogram equalization, you see that the calcifications are not clearly visible. If you see only that we are applying our non-linear function, which we have done previously, calcifications are visible, but still they are not very sharp. But now if you see, we have used this non-linear function in the unsharp masking, you see a very sharp image has come where these dots are now very clearly visible. So you see that that non-linear transformation function, when we have hybridized or superimposed it with the unsharp masking model, you can see the change in sharpness. Earlier results that you have seen, those results were good enough for your masses, but they were not very good for your calcifications. But when unsharp masking is applied with this nonlinear function, you see a very sharp response has been obtained over here. So when such sharp responses are available, one do not have to go for an independent segmentation approach. You can directly segment your output just by a small thresholding process as it's shown over here. So for further study in this, you can refer to my paper, which is published in Unsharp Masking Approaches for HVS-based Enhancement of Mammographic Masses, a Comparative Evaluation. This paper of mine is published in Future Generation Computer Systems. And you know, it compares how mammographic masses can be tested with different types of unsharp masking approaches. And you will come to know that when you apply this non-linear kind of approaches for and you are merging it with the unsharp masking, the responses are very, very much improved. So over here you can see that different types of unsharp masking approaches are applied, and you are again combining a non-linear function. A non-linear function you are combining over here in your existing unsharp masking module. So this is how that non-linear function can be used with some other approach in unsharp masking. Now another thing, there is another approach, there is another sphere where one of my students have worked in this domain, and that is that, you know, if you saw the earlier part, the sigmoidal function, we were doing manual tuning of parameters. Now this is another work where multi-objective optimization has been applied over here. Genetic algorithm has been applied over here, so that what we were doing in the form of the manual tuning over there, we are now trying to do it with the genetic algorithm, so that, you know, a more optimized set of parameters are available for this new sigmoidal function. And, you know, this only just you can see that the modification of this sigmoidal function and its optimization, only this much. I'm not done any CAD, I have not done any CADX, and just, you know, this improved transformation function with optimization with genetic algorithm, this part has recently been accepted in IEEE Census Journal again in this month only, in the month of September. So you can see that the proposed mammogram model exploits locally the properties of the sigmoidal function based on multi-objective genetic algorithm. So only the same transformation function is used, and we have tried to just optimize its values for multi-objective with multi-objective genetic algorithm, and we have applied the results on the mammograms. So this is the paper, and I think very soon, in another 1 week to 15 days, this paper is going to be coming online, and that is, it is dealing with the improvement of the local S curve. So your S curve is nothing but this is your sigmoidal curve, and you are using genetic algorithm over here. So I'm sure these have been the different avenues how you can go in for different types of research problem formulations by simply modifying different approaches, conventional approaches, and applying it for computer aided analysis. So I'm concluding the session with this. Any questions from anyone? Hmm.
Hello, sir.
Yes.
Sir, good evening, sir. I'm Sirisha, sir.
Okay.
From Guntur, sir. There is one doubt, sir.
Hmm.
So, can we classify images, that is the MRI images or the breast cancer images, that whether they are malignant cancer or the type can we identify, sir?
Come again. Actually, I'm not able to hear you properly. There is some.
By applying image analysis methods, can we identify the type of cancer?
Yes, certainly, because, you know, once you are enhancing the particular image, you are doing what I have told, what I have explained, that you are enhancing the contrast, you are enhancing the edges, and once the edges are clear, then only, you know, a radiologist or a medical expert can easily discern that what is the status of affair of that tumor, that whether it is still benign or whether you see that the edges are being percolating, it is getting radiating out. So if the edges are radiating out, that means that now that benign cancer cells are getting, you know, more cancerous, more active, and the metastasis has already started. So that actually gives the idea of, you know, the particular stage where the cancer is.
Sir, actually means I want to know whether this what you have explained based on the type you can identify whether it is benign or malignant. I want to know whether this the same can be applied for MRI images also, brain tumor images.
See, brain tumor images, it can be applied, but again, there have to be some customizations over there. I mean, you cannot apply the same function directly.
Okay.
You have to make certain change of parameters over there because again, as I told you, that MRI images are, little clearer images. Mammographic images are very poor contrast images. So we are visualizing here from the point of view, that you are applying an algo on a poor image. But MRI images are comparatively better in view. So certainly the same function cannot be, directly replicated, but you may have to do few customizations over there. Maybe you have to change the value of the adaptive gain over there. Maybe you have to change the value of K and B over there, and then that thing can be experimented.
Okay, sir.
Okay.
Thank you, sir. Thank you.
Okay. Any other question?
Sir, one speaker have posted her question in chat box. I'll just read out. I need to formulate 3 or 4 objectives in my research area. In this case, then I will identify new or proposed methods, detection methods, feature extraction techniques, and classification algorithms and objectives, or only particular area like different processing methods.
See, it all depends that how depth, you are going. If you check out like this particular work, that you have, say taken up, you just try to visualize that how I have chosen the kind of research problem over here. We have taken up one processing method. We have improved it as a new function. So say this is one complete full objective, because, in this full research, one paper can be published, right? Now, if you are, say, going to optimize the parameters of that function with some optimization algorithm, you can go up with another objective with that, right? And then similarly, getting it? Third part is that, say, you have used this nonlinear function and you have tried to, combine it with, say, unsharp masking, or say you have tried to combine it with some wavelets or some multi-resolution approach, and maybe you get a good result in some different domain of abnormalities. So that becomes a different objective. So, it all depends that how you are framing your problem. If, say, one way is only simple, that you do one objective complete on your enhancement problem, and then you go with the routine feature extraction and computer this machine learning. So this is how, your interest, your literature survey, that how you can frame that problem.
Sir, can I take decom images? I think you have taken .jpeg images. Is there any difference between JPEG and .decom images?
See, all these databases have different category of images. If you go for a different databases, maybe some databases have PNG images, some databases have JPG images, some databases have decom images over there. So, with the database, maybe it is their storage mechanism, that particular image can be there. Only point is that, such transformations you can do at the stage of the pre-processing, that, you may convert the image from one format to the other based on your requirement at the pre-processing stage, and then you can go in, say, for your further processing in the form of enhancement and segmentation.
Sir, if you don't mind, can you switch on your webcam once, please?
Any other questions? Any other questions? Anyone? Yes.
How features can be trained in CNN?
Pawan, I can't hear that question, I think, completely.
How features can be trained in CNN? This is the question asked by Manjula ma'am.
Okay. See, again, that is, the internal training attribute of your, CNN toolbox. It has nothing to do with, you know, what I have discussed right now, that is in the sphere of enhancement or, you know, feature extraction. It is, you know, totally different aspect, which is, you know, applicable to a deep learner. So which is, you know, very, very specific to your toolbox, where you have automated methods are just available, and you can do the training over there directly on that deep learner toolbox, right? It has nothing to do right now with what I have discussed.
Sir, what pre-processing methods you will follow for histopathological images?
Sorry?
For histopathological images, what pre-processing you will be doing?
See, again, that particular part you have to go in and with the analysis of those kind of images, that what is the region that you want to enhance in that. So that particular algo you have to pick up over there. So it is, you know, with the change of the nature of image, you have to change that particular algo. Suppose, you know, for you go for a histopathological images, say suppose you have a microbial images, and where you want to, you know, focus on the periphery of the bacteria which are visible in those kind of images, right? So that particular thing you can do with your, say, unsharp masking methods could be very good for that purpose, right? Suppose you want to say very dotted or very fine things, very fine features you want to observe over there, then you can go in for a multi-resolution processing over there. That could be very good in such cases. Yes.
Any more questions from audience? Without ROI, can I do classification? This is the question from Ms. Kanya Kumari. Sir.
Yes, it is also possible that you have to develop such type of algorithms, you know, which can be so suited that they can work on your direct images over there without, you know, knowledge of the ROI. So if you just recall, I have mentioned one of my paper of IEEE Census Journal, that is the polynomial filters. So that particular filter is dealing with such type of images only, where, you know, the processing is not dependent on the region of interest, and it can be directly applied to the full mammogram image. So if you refer to that paper, polynomial filter paper of IEEE Census Journal, as I show in my slides, that particular filter over there is applicable for your full images. Without the knowledge of ROI, it can process. Any other questions?
Audience, if there are no questions, we can wrap this session up. Sir, Vikrant sir, it's our pleasure to have you with us, sir. Thank you for spending your valuable time with us and clearing the doubts of the budding researchers.
Okay. Thanks, Pawan. Just 2 slides more to show. And it is a call for paper advert that there are certain special issues where, you know, me and my collaborators are dealing with. So if anybody from the audience must be, say, interested and they are having some research papers in those domain, they can submit in those special issues. So one is the soft computing. It is an impact factor 3 journal, and it is soft computing in the defense system methods for application. Another special issue where the call for paper is still on from my side is complex and intelligent system special issue, right? Again, it is an impact factor 3 journal. Another special issue is specific to COVID and AI, and it is an Arabian Journal of Science and Engineering. So these special issues could be helpful for anyone, like if somebody is interested to post papers over there in that domain. There is one more journal, that is Recent Patents in Engineering. It is a Bentham Science Journal and Scopus Index Journal, where I'm in the editorial board. So a simple Scopus journal where its call for papers is again open and anyone can submit their research over there. Another journal, you have International Journal of Natural Computing and Research, where me and Dr. Nilanjan Dey both are editors, right? And again, this is a ACM Index journal. It's a DBLP journal where anybody who is having some research work ready, they can go in for submission over there. So that was all from my side. Thank you. And for any further queries, you can take down my email and we can get connected over there. Thank you so much.
Thank you, Vikrant sir, for sparing your valuable time and sharing your knowledge, sir. It is really a valuable session.
Thank you, ma'am.
We expect your cooperation further, sir. If any of such kind of programs are there, we would like to invite you for that.
It will be a big pleasure to be associated with such kind of a gathering and team, and we are Siddhartha College. Thank you, ma'am.
Thank you, sir.
Yeah.